Welcome to Camel for You channel. Today we will discuss chapter 1 plant transport in that part 2. Water is essential for all physiological activities of a plant and plays a very important role in all living organisms. Here protoplasm contain water and different molecules are dissolved in several particles are suspended. Watermelon contain over 92% water and herbaceous plants have 10 to 15% of water, fresh weight of a dry matter. Terrestrial plants take up huge amount of water daily, but most of it lost to the air through evaporation from the leaves. This process is known as transpiration. A mature can plant absorbs almost 3 liters of water in a day, while a mustard plant absorbs water equal to its own weight in about 5 hours. Water potential is a concept fundamental to understanding water movement. It is components, solute potential and pressure potential sum up with these two. Water molecules possess kinetic energy. In liquid and gaseous form, they are in a random motion, both rapid and constant. Here, greater its kinetic energy or water potential. It is obvious that pure water will have the greatest water potential. Water will move from the system containing water at a higher water potential to the one having lower water potential. This process of movement of substances down a gradient of free energy is called diffusion. Water potential is denoted by the Greek symbol psi and is expressed in a pressure units such as a pascals PA. Pure water at standard temperatures which is not under any pressure is taken to be a zero. This is a wrong or right sentences bit. All solutions have lower potential than pure water. The magnitude of this lowering due to dissolution of a solute is called a solute potential, denoted with the psi s. Psi s is always negative. The more the solute molecules, the lower psi s. If a pressure greater than atmospheric pressure is applied to pure water or a solution, its water potential increases. It is equivalent to pumping water from one place to another. Pressure can build up in a plant system when water enters in a plant cell due to diffusion, causing a pressure to build up against the cell wall. This makes the cell turgid. The magnitude of increment in potential, water potential, in such turgid cell called as a pressure potential. So, this potential is denoted as a psi p. Osmosis is the term used diffusion of water across a differentially or selectively permeable membrane. Osmosis occurs spontaneously in response to a driving force. The net direction and rate of osmosis depend on both the pressure gradient and concentration gradient. The potato osmometer experiment. While in school, tuber is placed in water, the cavity in the potato. The tuber containing a concentrated solution of sugar collects water out from the outside due to osmosis from semi-permeable membrane. So in this, A and B both are here showing solute molecules and water. Here semi-permeable membrane and a less or high concentration of solute. A demonstration of osmosis. A thistle funnel is filled with a sucrose solution and kept inverted in a beaker containing water. Here, water will diffuse across the membrane in A to raise level of the solution in the funnel. In B, pressure can be applied as shown to stop the water movement into the funnel. The plant cells which regard to the water movement depends on the surrounding solution. The external solution balances the osmotic pressure of the cytoplasm is said to be isotonic. If the external solution is more dilute than the cytoplasm, it is hypotonic, and if The external solution is more concentrated, it is hypertonic. Cells swell in hypotonic and shrinks in hypertonic. Plasmolysis occurs when water moves out of the cell and the cell membrane of a plant cell shrinks away from its cell wall. This occurs when the cell is placed in a solution that is hypertonic to the protoplasm. The water, when drawn out of the cell through the diffusion into the extracellular outside of the cell fluid, causes the protoplast to shrink away from the walls. Initially, this phenomenon is indicated by shrinkage of protoplast, leading to the separation of plasma membrane from the cell wall in this corner. This stage is called as incipient plasmolysis. When the cell wall is left in the hypotonic solution for more time, the protoplasm completely shrinks, whereas in the cell said to be the plasmolyzed. Cells are placed in the hypotonic solution. Water diffuses into the cell, causing the cytoplasm to build up pressure against the wall. This is called turgor pressure. The pressure exerted by the protoplast due to the entry of water against the rigid wall is called pressure potential, with the denoted psi p. Imbibition is a special type of diffusion when water is absorbed by solids collides, causing them to enormously increase in volume. The classical example of imbibition are absorbable water by seeds and dry wood. Imbibition is also a type of diffusion since water movement is along a concentration gradient. The seeds and other such material have almost no water, hence they absorb water easily. Water potential gradient between the adsorbent and the liquid imbibant is essential for imbibition.